Hello. Welcome to the Lymphoma Research Foundation's Update on CAR T Cell Therapy in Lymphoma webinar. I'm Robin, and I'll be your moderator for today's call. During today's call, you will hear from two expert speakers, and you will have an opportunity to ask questions. If you have questions during today's presentation, you can ask them at any time in the Q&A box on the webinar. If you're listening by phone, you will be able to ask your questions during the Q&A session. As a reminder, this webinar is being recorded. At the end of the program, a link will appear on your screen. Please follow this link to complete an evaluation of this program and gain certification of attendance. If you are listening by phone, this link will be sent to your email at the end of the webinar. I'm now pleased to introduce Jesse Brown. Jesse Brown is the Associate Director of Patient Education at the Lymphoma Research Foundation. Welcome, Jesse.
Thank you. And thank you to each of you for taking the time to join us on today's update on CAR T cell therapy in lymphoma webinar. We'd like to thank our sponsors of this webinar, Kite, a Gilead company, and Novartis. We have callers from 48 states across the U.S., as well as patients and caregivers from 9 countries around the world. Access to expert disease information is so important, and we are thrilled to be able to bring you this educational program. The Lymphoma Research Foundation is a trusted resource of evidence-based lymphoma information and support services for the lymphoma community. During challenging times such as these, LRF is in need now more than ever. We remain steadfast in our mission to eradicate lymphoma and serve those touched by this disease, and support from our donors make this possible. If you would like to support the Lymphoma Research Foundation as we work diligently to assist lymphoma patients, survivors, and their loved ones, and provide timely lymphoma-specific information and education as it relates to COVID-19, please visit lymphoma.org/donate to make a gift today. We have a wonderful program planned for you today, and I'm honored to introduce you first to Dr. Krishna Komanduri. Dr. Komanduri is a professor of medicine at the University of Miami Miller School of Medicine. He is also the Chief Division of Transplantation and Cellular Therapy at Sylvester Comprehensive Cancer Center. Dr. Komanduri, I will now turn the talk over to you.
Thank you very much. It's a pleasure to be here, and I want to thank the Lymphoma Research Foundation for not only hosting this webinar, but all it does in the space of patient education and advocacy, as well as really raising the issues of novel therapies and research for patients with lymphoma. And I want to say it's a pleasure to be here with also my colleague, Dr. Jacobson from Harvard Medical School. So I'm actually going to work with Dr. Jacobson today to really cover, I hope, a number of topics in this really complex and exciting arena. And because of, I think, the broad range of topics here, we're going to try and keep this at a relatively high level and also leave enough time for conversation and discussion from individuals who want to know more. So forgive that first, and the things that I'm going to talk about in the beginning of this presentation are really providing an overview of what CAR T cell therapy is and discussing some of the benefits of CAR T cell therapy for lymphoma in particular, and start to address issues related to the side effects of CAR T therapy, and then I'll turn it over to Dr. Jacobson for further thoughts and discussions, including additional implications that you'll want to know about in future directions. So first, what is CAR T cell therapy? And I think I'm going to start there by talking about how we've traditionally approached lymphoma. So lymphoma, as you know, or most of you know, is a cancer of lymphoid cells, and lymphoid cells are really cells in our white blood cell system that typically live in either the bone marrow, but largely in lymph nodes, which are healthy little organs that can swell. For example, when we get glands that swell in our neck, when we get a sore throat, those are healthy lymph nodes that are swelling. In the context of cancer, the normal white blood cells that normally replicate and, again, provide our immune responses can become cancerous and then grow in ways that are unrestrained and indeed can disseminate throughout the body and cause fatal complications if really untreated. We approach the treatment of lymphoma the way that we approach many diseases, and that's typically to use combinations of traditional chemotherapies that basically affect dividing cells. They affect the cancer cells the most, but they also have important implications on the function of healthy cells, especially cells in our normal immune systems, and can leave patients vulnerable, for example, to infections and low white blood cell counts after treatment. We've known for decades that there are other approaches that we realized under certain circumstances were non-cancer chemotherapy based, but could lead to improvements in cancers. And some of those approaches include ways to basically use human immune responses to treat cancers, which is something that we've always had an interest in doing, but there have been relatively few ways of doing this. One of the first approaches that we recognized we could use to treat cancers were a form of stem cell transplants, where we actually give patients chemotherapy and give them white blood cells and bone marrow cells, what we historically called bone marrow transplants, from another individual. While we thought that that was an approach that really depended most for success on the high dose of chemotherapy, we also recognized that that is a way of giving white blood cells from a donor that actually eliminate cancer cells that survive the chemotherapy in recipients. There are also selected approaches, including giving proteins that are normally made by white blood cells, like interleukin-2 or interferon, that had a small role in treatment of a subset of cancers. More recently, we recognize that there are a subset of cancer cells that are, that are responsive to white blood cells that can be modulated by a class of drugs called checkpoint inhibitors. And there are solid tumors, including melanoma and lung cancer, that can respond to these drugs that effectively take the brakes off normal immune responses and can lead to improved responses that can be lasting in a subset of individuals. But now we really have, I think, with CAR T cells, a completely different approach, where a patient's own healthy white blood cells are actually taken outside of the body and modified and basically made into almost smart bombs that can treat cancers that have failed to respond to chemotherapy. For lymphoma, the traditional way of giving combinations of chemotherapy can cure a large fraction of patients, perhaps 60 to 80% of patients, depending on the stage and the risk factors for an individual. But when those initial attempts to put the disease into remission fail, either because the patient doesn't respond at all, or that the patient does have what we call a complete response, that is, the disease becomes undetectable, but then relapses over time, the likelihood of secondary approaches treating the cancer successfully decrease. And if a patient relapses the first time, there is a subset of patients who can respond to another set of chemotherapy drugs. And if they respond, they're typically treated with high-dose chemotherapy and a stem cell transplant using their own cells. But if stem cell transplantation fails, or if patients fail to respond up front, the chances of responding to additional lines of therapy go down very significantly. So for patients who have multiply relapsed or what we call refractory lymphoma, patients have less than a 20% chance typically of responding meaningfully to additional lines of therapy. And the likelihood of their disease going completely away can vary, but for many individuals is less than 10%. So these are obviously, results that as clinicians leave us very frustrated and mean that for most of those patients, they will end up dying of their lymphoma, and that can happen in a matter of weeks or months. So CAR T cells are a way of taking our white blood cells called T cells, which are critical in our response to viruses and other pathogens in our environment, and actually modifying them so that they have a new receptor, and that's called a chimeric antigen receptor, because it actually takes elements of what we make in our own body, that is an antibody response, and puts effectively an antibody on the surface of a T cell with a receptor that's genetically engineered. So in this approach, just to give you a broad sense of how it's done, white blood cells are removed from a patient with cancer. Those cells are then sent to a central laboratory. Typically, there are some individual centers that are doing this in research trials at their own centers, but for the commercially approved therapies, those are sent to a central laboratory where the white blood cells from the patient are then genetically modified to have this new CAR T cell receptor that recognizes a target present on the surface of lymphoma cells and also some leukemias that express the same target, which is called CD19. The cells are genetically modified and expanded outside of the body from a number of cells that's collected to many thousand-fold or even million-fold greater in terms of numbers. And then those expanded cells are returned to the site of care for potential infusion. We prepare the patient for infusion of those CAR T cells by giving 3 days typically of chemotherapy that's generally relatively easy to take and effectively makes room in the body for those T cells to expand. Again, it's typically delivered in the outpatient setting. Most patients with CAR T cell therapy will receive therapy in the inpatient setting. That is, the cells are infused back into the patient over typically just minutes, though a subset of individuals who are thought to be having the right product and are at lower risk can be done in certain outpatient settings. But the vast majority of patients treated to date have been treated in inpatient settings. Those cells are then infused, and now those T cells have, again, this new chimeric antigen receptor that effectively acts like a GPS that allows them to hone in and destroy a cancer cell expressing that surface target just the way that it would normally respond to a virally infected cell in our own bodies. That is the way our normal immune response would act. Now, the important thing is that in clinical trials of patients who have had these high-risk relapsed or refractory lymphomas, the responses have actually been dramatically higher than what we would have expected for additional lines of chemotherapy. For example, in the Zuma 1 study, which looked at patients who had, again, multiple lines of therapy with lymphoma, the response rates overall were in the vicinity of around 80%, with about 50% of the patients treated actually having a complete response. That is, at the next assessment, patients had no detectable disease by traditional scans. Now, not all of those patients retain their responses, but critically, of the patients who are treated, the studies now have followed patients long enough that about 40% of patients, roughly, at 2 years, continue to be disease-free without any evidence of lymphoma. And those of us who are experts in lymphoma know that those patients at that stage are probably unlikely to relapse. That is, those patients are highly likely to be cured. So we have cure rates in the vicinity of approximately 40%, which is, again, much higher than what we would have expected, and probably typically less than 10%. So there were critical trials. The Zuma 1 study looked at a drug called oxycaptogene ciloleucel, and a second study from individuals at the University of Pennsylvania and others looked at another product called tisagen leclucel, which was also found to have quite similar response rates and complete response rates and likely cure rates. And this led to FDA approvals of these two agents, oxycaptogene ciloleucel and tisagen leclucel, and the commercial strategy by the two companies that have developed these products, Kite Gilead and Novartis, to implement broad number of centers across the country where these products can be received by patients who meet these criteria. That is, they have relapsed or multiply refractory lymphomas. The critical thing is that since FDA approvals, there are actually a broader range of patients who have been treated with these therapies. And in many cases, about half of the patients who have been treated in what we have termed real-world studies actually would not have met the criteria for the initial trials. That is, that they're often sicker, maybe older, and have laboratory values, for example, that would have excluded them from the clinical trials. And yet, now with closer to 3,000 patients treated overall, we have similar response rates in patients who are treated in the commercial setting to what was seen in the clinical trials, despite, if anything, a higher number of patients with slightly more advanced and more aggressive features. There are a number of studies, and Dr. Jacobson, my colleague, has actually been one of the individuals who has been a leader in this field, and I'll certainly let her comment further. But I think that some of the key things that we know is that there appears to be no upper age limit at which patients don't respond to these therapies. And in fact, there are some studies that suggest that the response rates may be equal or even slightly higher in older individuals. We do know, of course, for this and other therapies that older individuals who have other kinds of, again, chronic diseases may not always be as easily treated and may have higher risk of side effects than other patients. But this is something that is important, is that patients should not think that there's an upper limit for consideration. And we know that individuals, for example, in their 70s have received and benefited from these treatments. There are side effects from these treatments, and that perhaps is not surprising, because if we think about what happens when we get, for example, the flu, we can develop side effects like high fever or inflammation that causes chills and sometimes severe and potentially life-threatening complications. And in some cases, in addition to the effects of viruses on our own bodies, those side effects are because of our immune systems and our white blood cells actually expanding, responding to the flu. And in this case, white blood cells that are genetically engineered to treat lymphoma can also expand and actually cause inflammation. So the vast majority of patients treated will develop complications, including fever, and a subset of patients will develop severe inflammation that can actually cause them to have shortness of breath or low blood pressures, and a subset of patients required ICU monitoring. In addition, there are complications that we don't understand very well with respect to why they occur, but they include effects on the brain, including changes in the way that patients think and sometimes can communicate. Some patients have difficulty finding words and actually sometimes cannot communicate at all. Others can develop severe complications, including seizures and coma-like conditions. The good news is, in the last 3 years, clinicians have understood how to recognize these signs and symptoms of toxicity. Management algorithms and approaches have been developed, and we know how to initiate often supportive care measures that limit the severity and the seriousness of these toxicities. So in some cases, they can become quite serious, and there are patients in all of these trials who have died from these complications. However, I think the important thing for patients to know is that the relative likelihood of having a serious or fatal complication from the therapy themselves is greatly outweighed by the benefit for patients, especially who have serious and refractory diseases that are often unlikely to respond. So there are approaches that I think should be discussed with any clinician, and I think we all know in our academic centers to both present the benefits of these therapies with the appropriate side effects. So those are some of the key issues. I would say, to summarize, CAR T cells are a novel way of taking our healthy immune cells, genetically modifying them, expanding them outside the body, and infusing them, and that in the trials that we have to date, they have dramatic and really, I think, revolutionary response rates relative to prior therapies. But these are novel therapies that have largely been applied in academic centers with great experience in treating lymphoma and leukemia, often also centers that are very experienced in stem cell transplants, and therefore have very experienced individuals from the laboratory to physicians to nurse practitioners and pharmacists and intensive care unit staff and neurologists that specialize in taking care of these novel complications. So this is not something that can be, we believe right now, appropriately delivered in many community oncology centers yet, though it's possible that, again, with evolution in the field, that will be possible in the future. Largely, it's now confined to typically 50 to 100 academic centers in the country where we think we can both make the decision about who to treat, how to manage these potential side effects, and also manage, again, the long-term issues with patients who both respond and don't respond to these therapies. So I'm going to stop there. I'm going to turn it over to my esteemed colleague, Dr. Jacobson, and look forward, along with her, to questions at the end of our formal presentation.
Thanks, Krishna. So my name is Karen Jacobson. I am an assistant professor of medicine at Harvard Medical School and a lymphoma doctor at the Dana-Farber Cancer Institute in Boston. I also lead our immune effector cell therapy program, which is largely consists of our CAR T cell program here. And so I'm going to pick up where Dr. Komanduri just left off, thinking about some of the side effects of CAR T cell therapy. And he talked a little bit about the inflammation that the T cells can cause, leading to a syndrome that we call cytokine release syndrome. And if that were to happen, that usually happens within the first 1 to 4 days after the CAR T cell infusion, while the patients are undergoing very close monitoring at the treatment center. Often that monitoring is inpatient, but there are some examples of these products being given as an outpatient. So, for instance, Dr. Komanduri discussed tisagenlecleucel, or Kymriah, which is the Novartis product, which has been tested and given in the outpatient setting. And we are anticipating the FDA approval of a third product, a third CD19-directed CAR T cell product for the treatment of fast-growing B-cell non-Hodgkin lymphomas, and that's called lisocabtagene maraleucel. And that product actually was formally tested in an outpatient setting, and a number of patients were able to be treated outpatient, and actually a good proportion of patients were actually able to stay outpatient through the entire follow-up period. The risk of cytokine release syndrome really kind of peaks when the CAR T cell expansion peaks, and then symptoms generally subside as CAR T cell numbers go down over time. And so typically people experience the side effects of this anywhere from 3 to 5 days. And if it were to get too extreme, we can dampen down some of the inflammation with anti-inflammatory medicines that really target some of the key mediators of this inflammation, as well as with steroids. Towards the tail end of cytokine release syndrome is when that neurologic toxicity that Dr. Komanduri referenced can arise, and not everybody gets this, but people who do get it, it can range anywhere from sort of just mild confusion or disorientation to really an inability to do what we call activities of daily living, like get out of bed to go to the bathroom, know what to do with a fork and a spoon to feed oneself. My favorite test is to give someone their cell phone and see if they know how to turn it on and dial a number. And so, and the most extreme cases are when people get very, very sleepy, and it's hard to get them to wake up. Again, as Dr. Komanduri alluded to, we don't totally understand the mechanism of this, but know that it does involve some amount of inflammation of the brain, and we typically treat this with steroids. But like cytokine release syndrome, this will subside with time and is almost uniformly reversible, and people will get back to their baseline cognitive status and mental status. And if people get this, that time to recovery can be as short as a couple of days, but can be longer, and sometimes it can take a couple of weeks for that inflammation to subside. But when people do leave the hospital after CAR T cell therapy, or people are sort of out of the window of needing close observation, it's really unlike stem cell transplant, where which more people are familiar with, where patients have a set of precautions that they need to follow. The only precautions really that we advise patients after they are treated with CAR T cells, we ask them to be within a 2-hour radius of the treating center for up to 4 weeks following the infusion, and that's because some of these side effects can recur. It's rare that they do, and when they do, it's generally mild, but we want them to be in close proximity to the treating center that has an expertise in dealing with those side effects. The other thing is that we ask people not to drive or operate heavy machinery for up to 8 weeks after CAR T cell infusion. And the reason for that, again, is that these symptoms can recur, and we're particularly concerned if the neurologic symptoms recur while someone's driving or operating heavy machinery. And so that was something that the FDA had asked be part of the approval for these agents. In terms of ongoing long-term side effects and long-term precautions, there are none. In terms of precautions, in terms of the side effects, things that we watch out for are low B cell numbers over time. So these CAR T cells obviously target the cancerous B cells, by targeting CD19, but our healthy B cells, which are part of our immune system, also have that protein on their surface. And so for some time after the CAR T cell infusion, the healthy B cells will decline in number, but we know that patients can survive with diminished or even absent B cells. And we know that, and we protect them from infection by giving them back the proteins that B cells typically make to help protect people from infection, and these are called immunoglobulins. And so we measure immunoglobulin levels in patients who have had CAR T cells over time, and when they fall below a certain level, we often preventatively give them an infusion of immunoglobulins to help boost their immune system and protect them from infection. About 20 to 30% of the time, patients can have their blood counts dip after they leave the hospital or after they finish their observation period following their CAR T cell infusion. And the mechanism for this is not entirely well understood. When this happens, we can help boost the white blood cell count, often with growth factors like Granix or Neupogen that you might be familiar with. And we sometimes need to give ongoing blood and platelet transfusions. For many patients, this will get better with time, but that sometimes can take months or even a year to get better. So that's where we are in terms of potential treatment side effects and follow-up care after patients leave the hospital. And I think a lot of where the field is going is recognizing that this is really the end of the beginning. This has definitely been a revolution in the treatment of lymphoma patients and oncology patients across the board, but we are really at the very beginning of maximizing and optimizing these therapies, and I think there's a lot more that we can do. So a lot of the questions that I've been seeing on the website have focused on, what other lymphomas are CAR T cells being investigated to treat. And so I'll touch upon that for a second. So as we've already alluded to, the approved products were tested in the fast-growing B-cell non-Hodgkin lymphomas, so things like diffuse large B-cell lymphoma, high-grade B-cell lymphoma, transformed lymphomas that have transformed out of some of the slower-growing lymphomas, and a disease called primary mediastinal large B-cell lymphoma. But the CAR T cells have been tested in mantle cell lymphoma and follicular lymphoma and are being tested in marginal zone lymphoma as well. Those are all B-cell and non-Hodgkin lymphomas. And we heard the results of the Zuma 2 study from Kite, looking at a CAR T cell product that's a little bit different than Yescarta or axicabtagene ciloleucel, only in that there's an extra step once the cells are collected to make sure that there are no lymphoma cells within the cells that are collected before they undergo the manufacturing process. And the results of that study are amazing. This is a group of patients that have failed ibrutinib or acalabrutinib, some of the BTK inhibitors, and treatment options have been fairly limited for those patients. And the response rate to KTE-X19, which is the CAR product that was tested in the Zuma 2 study, was over 90%, with 67% of patients having a complete response. And so median follow-up on this study right now is 12 months, and the majority of patients who have a response are still in response at the 12-month time point. And so because of that, Kite filed for FDA approval for that product for mantle cell lymphoma. And so one of the questions was, when do we think it's going to happen, and do we think it's going to potentially be delayed by COVID-19? And I can't answer that question. I think none of us really understand the complexity of what the FDA may be dealing with right now, but we certainly anticipate or are excited for the potential for that product to be FDA approved before the end of the year. Kite also had a study in both follicular lymphoma and an ongoing study in marginal zone lymphoma, and this is all patients who have had at least 2 prior lines of therapy. And so the follicular lymphoma part of that study has been fully accrued, and we're awaiting the results. We're waiting the follow-up to mature long enough to be able to see the results, but we're hopeful that that's going to provide a new treatment option for patients with relapsed follicular lymphoma, and the marginal zone lymphoma study is ongoing. There are questions that have come up that said, do we really have to wait for patients to be refractory to chemotherapy? And that's being addressed in 3 different randomized studies from the 3 different companies that have CD19 CAR T cells. So that's being addressed by Kite in the Zuma 7 study, where they're taking, and it's being addressed by Novartis, as well as by Juno and Celgene. And these studies are all very, very similar. So they take patients with diffuse large B-cell lymphoma who get standard frontline chemotherapy, and if patients either don't enter a remission to that chemotherapy or relapse within the first 12 months of that chemotherapy, they then get randomized to either getting CAR T cells directly or getting second-line chemotherapy followed by an autologous stem cell transplant if they respond to that second-line therapy. And so we await the results of those studies to see if possibly moving CAR T cell therapy up into an earlier line of therapy may make a difference. There was a question about T-cell lymphomas and whether we could use T cells to combat cancers of T cells. And the answer to that is yes, there are trials looking at using T cells to fight T cell cancers. You have to do some additional gene editing or editing of the cells in those cases, because you don't want the T cells that you're engineering to target other T cells to also target themselves. So you're going to have to remove the target from the engineered T cells before you give them back to the patient. But there are trials that are ongoing with that. And people are also looking at CAR T cells for Hodgkin lymphoma, targeting a protein on the surface of those cells called CD30. And those trials started at Baylor and at the University of North Carolina, and they're entering phase 2 studies in a larger patient population across the country right now. So that's how the available CAR T cells are expanding into other lymphomas. And also new CAR T cells are expanding into new types of lymphomas. But I think we can make these CAR T cells even better. So we talked a little bit about the fact that 60% of people still are not being cured with these CAR T cells. We also talked about the side effects. And so what can we do to make this better? So one is to understand why some people aren't responding. And there are two main hypotheses that are being worked on right now. One is that the T cells get exhausted, that you give them back to the patient, they get super activated, and they basically get exhausted. They run out of steam, and that's related to interactions between markers on the T cell with markers on the tumor or the cells surrounding the tumor. And so that type of exhaustion can be overcome by combining some of these CAR T cells with some of the other immunotherapy drugs that we've heard so much about in recent years, things like pembrolizumab and nivolumab, and these are the checkpoint inhibitors that maybe can keep the CAR T cells active and engaged for a little bit longer. And another mechanism of resistance is that the cancer cell essentially loses CD19, so that they no longer are a target for the CAR T cells. And so options to overcome that mechanism of resistance is to actually create a CAR T cell that doesn't only target CD19, but targets other proteins on the surface of the tumor cell. So, for instance, in some of the lymphomas where CAR T cells are being developed that target both CD19 and CD20, in some of the leukemias, CAR T cells are being developed that target CD19 and CD22. So that's another mechanism. The other thing to realize is in terms of the side effects of these CAR T cells is that right now these CAR T cells get turned on and they're just on, and there's no sort of modulation of them. There's no way to control how many of these receptors for the tumor are on the surface of the T cell or how activated the T cell gets. And so I think this is, although this seems so innovative and so revolutionary right now, we'll look back on this 5 or 10 years from now and think that we were really doing this in sort of a kindergarten kind of level. I think there are a lot of things that we can do to really modulate the T cell response so that it's more like what happens when we are responding to a flu or some other virus. And so ways that people are looking to do that are having extra switches in the T cell that we give back to the patient. So some new CAR T cells require, an interaction with one protein on the tumor cell to turn the T cell on, and then the T cell doesn't get really activated unless there's a second interaction. So it requires two signals for the T cell to turn on. And then there are other T cells that are being developed that if it gets turned on too much from one interaction, there's some mechanism by which the T cell will shut itself down. And then there are also engineered T cells that are now being developed where all the signaling actually goes through the native T cell receptor. So rather than getting this, super excited signaling through this, novel protein on the surface of the cell, that novel protein interacts with the T cell receptor, and the T cell gets activated in the normal way that it would normally get activated. So these are all new ways to think about the T cell and how to modulate it and how to control the amount that it gets activated, so that potentially we can not only make these safer, but also avoid the T cell exhaustion that happens, and thereby make them potentially more effective. And then finally, we couldn't talk about sort of new options or new products for treating patients without talking about the allogeneic CAR T cells. So all of the products that we talked about today are all involve collecting T cells from the patient themselves and then engineering those T cells and giving them back to that same patient. But this has some downsides. So one downside is that these T cells are coming from a patient who has a blood cancer and who has also had chemotherapy for that blood cancer before. And so they may not be the most healthy T cells that we can imagine using to launch an anti-tumor immune response. Another downside is that it takes 3 to 4 weeks to make these T cells in the lab, and so that the patient with the lymphoma that's not been successfully treated has a lymphoma that's progressing during that time, and that can be very rate limiting for some people. And then finally, and one of the questions we had was that, CAR T cell is so expensive. And so a lot of that has to do with how complex it is to collect T cells from one patient, send them to a laboratory, have them be engineered over 3 to 4 weeks, and then sent back to the cancer center. And so the donor T cell, if we're going to be able to use donor T cells, it gives us the opportunity to collect from one collection to potentially make 100 different products and treat 100 patients, which could cut down on the cost. And so allogeneic CAR T cells are entering clinical trials. The way that they're engineered so that they don't attack the patient in an unforeseen way is that the T cell receptor is actually deleted from the T cell surface, so that these T cells can only recognize CD19 on the lymphoma cell. They can't recognize the other cells within the patient's body. The problem is that those T cells have markers on them that belong to the donor. And so when we give them back to a patient right now, the patient's own immune system may reject those T cells. And that's been what's been rate limiting so far for these donor or allogeneic CAR T cells. But the hope is that we'll figure out how to either dampen the host immune response, the patient's immune response, so that these T cells can exist long enough that they can kill the cancer cells, but also, or do something to the T cell, the engineered T cell itself, to avoid the host immune response. So I think, those are the new modifications that are going on in the laboratory and hopefully will translate into new clinical trials going forward. So I think we're nearing about 40 minutes. There are a lot of questions that I can see already being entered from the audience. And I think that I will end with just really emphasizing the fact that this has been a revolution in oncology, and it's one that's here to stay. And we've really made a big impact in diseases that had no other options. But we're really at the end of the beginning, and I think the field is going to grow exponentially. In 2020, I think there are something like 300 CAR T cell trials open across the globe. We're expanding beyond the lymphomas that we have talked about today, and into other blood cancers as well as into solid tumors. And I think that we're really just at the tip of the iceberg here, and it's a wild ride, and we're excited to be on it. And so with that, I will stop, and then we can take questions from the audience.
Great, thank you both so much. Before we open the program to questions, I want to share information with you on the Lymphoma Research Foundation that may be helpful to you as you navigate lymphoma and long-term survivorship. The Lymphoma Research Foundation, LRF, is the nation's largest nonprofit dedicated exclusively to lymphoma. Our mission is to eradicate this disease through investment in the most promising lymphoma research and to serve those impacted by lymphoma through quality education and support opportunities. As we continue to make progress in advancing lymphoma research, we also want to ensure that you have access to the latest information about your disease. The Foundation provides comprehensive disease and treatment-specific resources, programs, and services, all of which are offered free of charge and have been reviewed by lymphoma experts. Most relevant to today's call, LRF offers a variety of lymphoma-specific resources, many of which you can access at the bottom of your screen if you're utilizing the web link, or via LRF's website at lymphoma.org if you are on your phone. The LRF helpline can answer your specific questions about lymphoma as well as discuss relevant treatment options in clinical trials. We also offer the Lymphoma Support Network, which is a one-to-one peer support program for people with lymphoma and their caregivers. The Lymphoma Support Network connects patients and caregivers with volunteers who have similar experiences to help give others strength to meet the challenges they may have to face. We also offer a variety of publications that have been reviewed by lymphoma experts to ensure you have access to the latest lymphoma information. We have comprehensive books on understanding non-Hodgkin lymphoma, Hodgkin lymphoma, and chronic lymphocytic leukemia. We offer a variety of easy-to-understand fact sheets on each of the subtypes of lymphoma, as well as supportive care topics that may be relevant to your lymphoma experience. We also have a dedicated CAR T cell therapy fact sheet and a CAR T cell therapy learning center. Finally, our mobile app, Focus on Lymphoma, is an award-winning app that provides patients and caregivers access to comprehensive content, as well as unique tools to help manage your disease. The app is available for free download in the Apple App Store and in Google Play, and I would encourage you all to download the app today. I really hope that you will take advantage of some of the great resources and services that the Lymphoma Research Foundation provides. If you have questions regarding what you learned today, or if you need information about relative treatment options and supportive care resources, you can reach out to the Lymphoma Research Foundation through our website at lymphoma.org or by calling our helpline at 1-800-500-9976. Thank you all again for your time today. We will now begin our Q&A portion of the program. Just as a reminder, please keep your questions as general as possible so that the entire audience can benefit from their answers. In addition, we are doing a webinar tomorrow related to the coronavirus and what the lymphoma community needs to know. If you have any questions related to that topic, please tune into our webinar tomorrow. You can sign up at lymphoma.org/webinars. And with that, I will turn the call over to our moderator.
Hi. If you'd like to ask a question, please submit your question through the Q&A box on your screen. We will take as many questions as possible. But if you have a question that does not get answered, you can always reach out to the Lymphoma Research Foundation's helpline at 800-500-9976. And now we have a question from online. First question is, do you envision CAR T being offered for frontline treatment?
So this is Krishna Komanduri. I'll take a crack at that first. So I think the important thing is that the current approved products are approved for patients who have failed multiple lines of therapy, because we know that the benefits of CAR T cell therapies and the risks, which are real but modest, as Dr. Jacobson and I described, are outweighed by the benefits. Knowing that we can cure a significant fraction of patients up front with standard therapies, there's always the risk as you move experimental therapies up that you might actually end up with an approach that, although attractive in many ways, as we've described today, might actually end up being inferior to conventional approaches and/or then leave people unable to, again, once somebody has failed immunotherapy, it's possible that they may not actually respond to additional immunotherapies. So there are trials moving CAR T cell therapies up to earlier places, for example, at first relapse, for example, and there's no doubt that there will be, for subsets of patients who are particularly high risk, trials that look at earlier lines of therapy. But right now, I think it's important to note that the evidence is strongly in favor of their role in patients who are, again, refractory or multiply relapsed. But we just don't have data about, whether these will be better approaches for patients with earlier stages of disease. I'm happy to hear Dr. Jacobson's thoughts on that too.
Yeah, I think I totally agree, and I think that what's important to remember is the diseases that we're talking about, these fast-growing B-cell non-Hodgkin lymphomas, are cured, 65 to 70% of the time with our first-line chemotherapy regimens, and so that's better than 40% that we're seeing in the multiply relapsed setting with CAR T cell therapy. I think that there are definitely patients who we know are at higher risk and have potentially lower cure rates with frontline chemotherapy, but there are still cures being seen. And so to move something like this further, either to second line or first line, we really have to be able to identify patients that are not going to do well with chemotherapy, and we're not quite there yet. I think that over time we may be there, and we may be able to look at a patient's tumor and be able to say this tumor has a very low chance of being cured with chemotherapy, and that would be a group of patients that we can go straight to something like CAR T cell therapy for. But our science isn't there yet, and I think that's the goal, is to avoid unnecessary chemotherapy for those who won't benefit. But right now, what we have to remember is that the majority of patients will still benefit from frontline chemotherapy.
Thank you. I have another question. Is there any hope of having the cost for CAR T decrease from what it is now? The cost of treatment is a big barrier.
Happy to take that. I've been doing a lot in the healthcare financing arena with this, and so I think it's important to note that the prices for that the two companies have set for these products, are about $373,000 for one infusion of cells. And I think that there's an honest, thoughtful assessment about whether, because this is one infusion and not multiple rounds of chemotherapy and provides, often revolutionary advance in benefit, whether that cost is justified. We know that this is a highly personalized therapy, and it probably costs $100,000 or more to actually simply manufacture the cells. And also doing these trials can cost $10 million or more. But that's an honest question, and I think one that we're trying to understand. There are ways in health economics of kind of trying to assess various treatments and understanding how they vary in terms of their value with respect to how many years of life people are likely to get. But that's complex and evolving. The other issue is that I think for most people that private insurers are largely, as long as patients meet the criteria that the FDA established at the time of approval, largely likely to have no problems approving this. There are issues with state and federal payers, that is, state Medicaid programs and Medicare programs. I have worked with the American Society of Transplantation and Cellular Therapy and Lymphoma Research Foundation and other organizations to try and improve the reimbursement for that, because right now there are barriers. I have no doubt that as the technologies improve, especially, for example, as we can take cells that are not manufactured individually for a patient, and we are all trying to look toward eventual approaches that we call off-the-shelf approaches, for example. For example, maybe one healthy donor could generate and have cells manufactured that could be used for a number of other individuals, and therefore the individual costs per patient will decrease. There are a lot of technical and immunologic issues that we don't have time to get into today. So I am confident. I do think that the cost of these therapies do represent a barrier. And again, the issues with payers are unique. And I think we actually need patients and organizations and physicians to advocate for the kind of change and assessment that we need. But that's a great question, and I don't think we should discount it, because these therapies, although revolutionary, are not going to help patients unless we can find ways to pay for it, and also, not have to take away from other medical, catastrophes like the one that we're all dealing with right now.
Thank you. Our next question is, I have large B-cell lymphoma. How do I choose between Yescarta, Kymriah, or any other clinical trials?
That's a really good question, and certainly as a patient, you shouldn't be choosing on your own. It should be a discussion that you have with your physician. But I think there are some very important things to note from some of these clinical trials. And also some of the real-world experiences that we've seen with these products. So one is, they do seem that the products that we have available to treat diffuse large B-cell lymphoma do seem to be fairly comparable in terms of efficacy. So that it seems like the same number of patients tend to respond, and the same number of patients tend to have these durable remissions. There may be a slight difference. There may be some important differences, I should say, in terms of the side effect profile, but remember, as we went through the side effects for these CAR T cell therapies earlier, the side effects are rarely life-threatening and are almost nearly 100% reversible. And so I think when I have a patient, we are able to give both Kymriah and Yescarta, and we have open clinical trials at our center. And when I have a patient in front of me, I often have to think about not only what is the safest and most effective product I can give to a patient, but which product am I most likely going to be actually able to treat the patient with. And on the Zuma 1 clinical trial with Yescarta, 91% of patients received their CAR T cells. On the Juliet study with Kymriah, there were about 50 patients that did not go on to get their CAR T cells for a variety of reasons, some of which were because the cells that were manufactured were not suitable for reinfusion, and some of it was because, given the time it took to manufacture the cells, patients got too sick to be able to receive their cells. So it's very important for me as a treating physician to pick a product that I feel is most likely going to come back and be able, and I'm going to be in a timely manner, and I'm going to be able to give that product to the patient. And that changes week to week. And so that's what I mean by saying it really needs to be a discussion with your healthcare provider. Obviously, we come from academic centers that are really invested in testing the newest and next best thing. And so but we all recognize, as we think about new clinical trials for diffuse large B-cell lymphoma with new products, we're all constantly thinking about, is it ethically okay to offer this to someone when we have FDA-approved products where we know very clearly what the efficacy and safety is? And I can say here at Dana-Farber, we only open clinical trials that we believe will have at least the same efficacy and safety profile as the commercial products. So it's a little bit of a leap of faith for a patient to trust that if a clinical trial is being recommended to them or being discussed with them, that it is felt to be as safe or as effective. But there are a lot of instances of ongoing clinical trials of CAR T cell therapy in diffuse large B-cell lymphoma where the rationale for the trial is very, very justified, and there's enough information to think that it's likely to be as effective. So I think that again is a personal discussion with your treating provider, but at least just have faith that if they're discussing those clinical trials with you, it's because they think that these may be as safe and as effective as the commercially available products.
Thank you. Our next question is, will the FDA approval of KTE-X19 CAR T cell therapy be delayed in light of the COVID-19 pandemic?
So I had addressed that.
Oh, yeah, go ahead. Go for it.
I was just going to say I had addressed that a little bit because I had seen that question before I started speaking, and had said that we anticipate the approval to be sometime this year, and whether my estimation, and I'm not sure if Dr. Commentary has additional insight, but my estimation was I really can't imagine what the FDA is encountering right now, and whether that bleeds into ongoing review of trials for consideration of approval. But I'm still optimistic that we'll see that be approved sometime this year. But let Krishna give his opinion.
Yeah, no, I was just going to say I don't think there's just so much moving so quickly right now that I don't think we can say, and I would share the optimism that obviously for critical therapies where we have the clinical trials completed that the FDA will continue to keep moving forward. But I would just say stay tuned. There's so much changing week to week that none of us know, and I hate to speculate further.
Thank you. Our next question is, CAR T cell therapy approved by insurance if I'm eligible to go through it?
So I'll address that again. There are the vast majority of commercial payers recognize the clear value of this therapy and the fact that despite, again, the significant cost, that it confers a very meaningful and revolutionary advance for most patients. Now, that said, they are grasping with, again, the unique costs and the way that these therapies are delivered. And I would caution patients actually to anticipate sometimes snags or even delays and sometimes missteps with respect to approval, and occasionally, unfortunately, this can even affect timelines where we are actually see a patient where we think that the therapy is appropriate and we have to wait for additional steps, for example, approvals of the therapy. So it is one of those things that I think that the one thing I would say from a patient perspective is not that you should stay up at night worrying about whether your insurance company is going to approve it, but this does reaffirm the need to get early referral to tertiary centers where these therapies are being approved, so that if there are delays or snags, that's less likely to impact your care or allow your disease to progress. So we can't say that those don't occur. They do occur at times. Many individuals have insurance companies that are, right on top of this and have developed novel systems. Others are lagging behind. So just know that there is a variable amount of understanding as these therapies come online, and there are new decisions and processes that are coming into place. But again, we will work with you. All of us have teams of individuals that specialize in the financial aspects and the approval aspects, and we try to dovetail that with, of course, the clinical decision making and the coordination of care with, for example, your lymphoma doctor, and so. But that can be an issue, and again, I would just say that the take-home message is make sure that you're seen as early as you can, and so that the impact, if any, can be minimized.
Thank you. I have one more question. Are there specific patient indicators that will help a clinician determine if a patient is a better candidate for CAR T than another treatment option?
I think when we're talking about patients that have relapsed or not responded to multiple lines of therapy, I think right now there's no other treatment option that benefits as many patients as CAR T cell therapy does. Another way to say that is that CAR T cell therapy has really become a standard of care for patients who have relapsed disease after 2 or more lines of chemotherapy. We don't have good predictors right now to determine who the 40% of people are who are going to have those long-term durable responses, the ones that we hope are cures. And we're all working on that. We're all working to try to identify who are the patients that are most likely to respond, and for the patients who are less likely to respond, what are the reasons that they're less likely to respond, and how can we improve that? But right now, there's nothing that exists that looks better than CAR T cell therapy for the largest number of patients in this setting. So I hope that answers that question. I don't know if Krishna has some other thoughts.
No, I think that's an excellent response.
Thank you. We are taking no further questions at this time. I'd like to turn the conference back over to Jessie Brown.
Thank you again to each of you for joining us on today's call, and we hope you found the information both informative and also hopeful. We'd also like to thank our sponsors again for making this program possible: Bristol Myers Squibb, Kite Pharma, a Gilead company, and Novartis. Please remember, if you have any additional questions or would like to be connected with someone else who has been impacted by lymphoma, you can reach out to the LRF helpline at 800-500-9976. Also, at the conclusion of this program, you will receive an email prompting you to complete a program evaluation. I would ask that you please take a few moments to complete this brief evaluation, as these are important for helping LRF to ensure they deliver the most useful and meaningful programming to you. And with that, I would like to thank you all again for joining us, and have a wonderful day.